cell cycle consists of three parts: interphase, mitosis, and cytokinesis. In the first part of interphase, in G1, the cell goes through some growth and does protein synthesis. In the S phase, the DNA replicates, so it changes from a single chromosome into an X, or a replicated chromosome. In G2, the cell grows some more and gets ready to divide in mitosis. In interphase, we see that we have a nucleus, a nucleolus, two pairs of centrioles to get ready for cell division, and the chromosomes are not yet formed, so DNA is in the form of chromatin. Prophase is the first phase of mitosis. You can see that the nuclear membrane and nucleolus have disappeared. The centrioles have moved to the poles of the cells, and they're anchored there by microtubules called asters. Spindle fibers form across the center of the cell, and the chromatin coils up into chromosomes. We have a cell with 2 of each chromosome, so 2N, with a total of 4 chromosomes in the cell. So 2N equals 4. In metaphase, the chromosomes line up in the middle on the metaphase plate. We can see that we still have the centrioles at the poles, spindle fibers are there, and we're going to remember M for middle. Chromosomes align in the middle of the cell, one above the other, getting ready to divide. We still have a cell with 2N equals 4 chromosomes. In anaphase, the centromeres divide. We're going to remember that they pull apart. So A in anaphase stands for apart. The reason they pull apart is that the centrioles pull in on the spindle fibers, shortening them. So now we have 1/2 of each chromosome on the spindle fiber. In other words, the sister chromatids now have become chromosomes. In telophase, we recognize it because the cleavage furrow has formed, the indent in the middle of the cell. Telophase has the opposite steps of prophase. The nuclear membrane reforms, the nucleolus reforms, and later in telophase, the chromosomes would decondense back into chromatin. Right now we'll show it as chromosomes to see that we still have 2N equals 4 in each daughter cell. So we started out in mitosis with 2N equals 4, and we still have 2N equals 4. At the end of telophase, we have cytokinesis. Cytokinesis is when the cytoplasm divides into 2, or we get 2 new cells. Each new daughter cell is identical to the mother cell, and so these 2 new cells are ready to enter the cell cycle again. The DNA has decondensed into chromatin, and now it can go through G1, S, where we have DNA replication, G2, and then mitosis.